This is going to be part 1 of chapter 8. We'll be talking about energy concepts, enzymes, and metabolism. So we're going to start with some just basic energy concepts. We'll talk a little bit just briefly about the role of ATP in cellular energy. We'll talk about enzymes. How do enzymes work? What are they? How is enzyme activity regulated? So if we're going to talk about energy concepts, we need to first define energy. And of course, energy transformations are something that is happening in every living cell and every living organism. And energy is defined as the capacity to do work or the capacity for change. So basically that ability to make something happen in a cell, in a living organism. And, since we're talking about this in a biological context, most of these energy transformations in living organisms, in living cells, are tied in with cellular chemistry, biochemistry. So that's kind of where we're headed with this discussion, is eventually we want to be talking about cellular respiration, some of the chemical reactions that are key for cells utilizing energy coming into the cellular environment. We want to talk about photosynthesis, how can plant cells capture energy into a chemical capacity inside of cells. So that's kind of where we're headed down the road with these discussions. You're probably familiar with the two main types of energy, potential energy and kinetic energy. Potential energy is stored energy, and again, in this chemical context, you're going to find potential energy stored in these macromolecules, carbohydrates, lipids, proteins, in the bonds that are holding those macromolecules together. You may remember in previous chapters we talked about covalent bonds, hydrogen bonds, some of the other types of weak chemical interactions. So all of those different types of bonds contain energy, and we can release that energy by breaking those bonds. Whereas kinetic energy is the energy of movement. So it could be something moving within a cell itself moving, or actual movement of an organism. So we see in this picture here this cat jumping up onto the counter, right? That's an example of kinetic energy. But of course that cat, ate its cat food or whatever it was fed. That cat food contained, sugars, carbohydrates, proteins, some of those macromolecules I was just mentioning. And those molecules, those bonds holding those molecules together, were being broken down inside the cat cells. And some of that potential energy was being transformed into that kinetic energy that's powering the muscle movement and the jump of the cat. Now, another general term we want to mention is metabolism. So metabolism is just defined as all the chemical reactions that are happening in an organism or in a cell at any given time. And there's really two main types of metabolism that you need to know. There's anabolic metabolism or catabolic metabolism. Anabolic metabolism or anabolic reactions are going to be reactions that are building larger molecules. So for example, linking two monomers together or multiple monomers together to build a polymer, that's going to be anabolic. And generally speaking, we need to put energy in to make that happen, and we're kind of making order from chaos in that type of anabolic reaction. Whereas in catabolic reactions, we're taking a larger molecule and breaking it down. And as I was just mentioning on the previous slide, we have stored energy in these chemical bonds. If we break those bonds through these catabolic reactions, we can release some of that energy and use it for other purposes. We also can't talk about basic energy concepts without mentioning some of the laws of thermodynamics. So thermodynamics literally translates as energy changes. These are laws of energy changes, and these are universal laws. And we're gonna think about these in terms of how these laws of thermodynamics would be applied at a cellular level in a living organism. So I want you to know the first two laws of thermodynamics. The first law, simply stated, says energy is neither created nor destroyed. So you can think of that as there's a constant amount of energy in the universe. We can't magically create new energy. We can't make some of that energy that's there disappear, but we can convert it from one form to another, as we were just talking about, from potential to kinetic and really back again. The second law of thermodynamics states that when you have these energy conversions happening, again, for example, from potential energy to kinetic energy, some of that energy becomes unavailable. And sometimes you'll hear people say, well, that energy is lost, but if we refer back to the first law, right, we can't really truly lose energy. We can just make it really unavailable for being able to do something useful. And we see this, in our own bodies all the time. If you're working out, for example, you tend to heat up, right? And so some of that heat is coming from these energy transformations, right? Potential energy in the sugars and fats and proteins in your body being converted into kinetic energy. Some of that energy becomes unavailable, gets converted into heat. And I'm saying it's unavailable because heat is just not very useful for helping carry out the work of the cell. So bottom line, the second law is saying, when you have these energy transformations, none of them are 100% efficient. So you're never going to get all the energy out of a macromolecule and convert 100% of it into kinetic energy. Some of it will be in the form of heat, and that's not very useful for us. Another term worth mentioning here is entropy, and entropy is just referring to the disorder or chaos in a system. And, as we're all aware, chaos, disorder tends to increase, right? Unless we make a constant concerted effort to keep things neat and tidy and organized, right? Things descend into chaos pretty quickly. And the same thing is true at the cellular level. If you think of a working, living cell, it may seem a little chaotic, but it's actually a very ordered, organized place. And so for a cell to maintain that order, really for a cell to stay alive, it needs a constant input of energy to kind of fight against that disorder, keep things neat and tidy and functioning correctly. And so a lot of this introduction is really headed towards this discussion here of what's called Gibbs free energy, which is symbolized by the letter G. And Gibbs free energy is something that cell biologists or biochemists would commonly determine for a specific chemical reaction. And we could define free energy, this Gibbs free energy, as energy that can be harnessed to do work in the cell at a specific constant temperature and constant pressure. So those two variables are always constant when a cell biologist is looking at free energy calculations. And so, in particular, what biologists and biochemists want to know for a particular reaction that's happening in a cell is what is the change in the free energy for that specific reaction. Is it a positive change? Is it a negative change? And that change is referred to as delta G. So, delta is always the mathy sign for change. And what that would tell us, if you know the delta G for a specific reaction and you know whether it's positive or whether it's negative, that would tell you whether that reaction consumes energy or releases energy. And for example, if you know that you determine that a specific reaction consumes a lot of energy, has a high delta G, then you could probably assume, well, that reaction may not be occurring very frequently because, the cell would have to use a lot of energy all the time to make that happen on a regular basis. Whereas if a reaction releases a lot of energy, maybe it does happen more regularly in the cellular environment. So the bottom line with changes in free energy with delta G is if a given reaction has a negative delta G, a negative change in free energy, that means that particular reaction releases energy. Whereas if a reaction has a positive delta G, that reaction consumes energy, requires energy for that reaction to occur. And again, as I was just saying, right, you could imagine if a reaction has a large positive delta G and there's, just not enough energy available in the cell at that time, then that reaction is not going to happen. Another way to think about this, this is showing a reaction that has a negative change in free energy, a negative delta G. Another term for this would be exergonic. We could call this an exergonic reaction. It releases energy. And this would be analogous, if you look down here, to the ball rolling down the hill, right? We don't have to put in energy to make that happen. Just due to the forces of gravity, that ball is going to roll spontaneously down the hill, energy being released. If we think of this in terms of a chemical reaction always begins with reactants and ends with products at the end of that particular reaction. And in this case, we're getting a negative change in free energy. So we have free energy here on the y-axis. And as this reaction proceeds, which is the x-axis is the time course here, we're seeing that the products have less free energy than the reactants. So again, bottom line, that reaction released energy, exergonic, negative delta G. And in this kind of situation, just like a ball rolling down a hill, these are things that are going to be likely to happen pretty regularly, right? If you see a ball poised at the top of a big hill, you can assume that ball is probably going to roll down that hill in short order. Same kind of thing with these reactions. If we know it has a negative delta G, we could assume as biologists it's probably going to happen pretty quickly. Whereas the flip side of that coin would be an endergonic reaction, a reaction that consumes energy, has a positive delta G, an anabolic reaction, if we want to go back to that metabolic term. And this would be analogous to pushing the ball up the hill, right? We do have to put in some energy to get that ball back up to the top of the hill. And likewise, if we want to think of this in terms of a chemical reaction, this would be a situation where the products have a higher free energy than the reactants. So we have a positive change here. And as I was saying before, anytime you have a positive delta G, energy has to be required. That reaction is probably less likely to occur as frequently in a cell. All right, so we've got our changes in delta G, and I'm not gonna have you calculate delta G's, and you don't have to memorize this equation, but I did want to just show you quickly what goes into determining a delta G for a specific reaction. And so, as I mentioned just briefly earlier, delta G's are always calculated in an environment with constant pressure and constant temperature. And so here's the simple calculation. So it's the delta H, change in enthalpy, which is basically the total energy in the cell, minus T, temperature, times delta S, which is the change in entropy, that disorder that I mentioned before. And you can probably imagine, temperature is pretty easy to calculate. Total energy might be more difficult to calculate. Change in entropy might be more difficult to calculate, but it can be done. And if you're a science major, if you end up taking cell biology or biochemistry down the road, you probably will end up doing some delta G calculations eventually. So I'm going to end part 1 here, and you can pick this up in part 2.